You guys are not gonna get no.
How are you doing?
Never.
How's everyone doing?
Oh, doing good.
Wonderful.
Yeah.
Okay.
Excellent.
Well, I have an interesting paper for you guys today. Okay, so I made, right, for all the new journal club members. Look, this is a cool paper, okay? It's by Zhang, right? At all.
It's our collaborator.
2019. Yeah, of course it's a collaborator, right?
really?
We'll find out real soon. And we're talking about the shell proteome, okay? So, protein in the shell that have to do with the shell and the regulation of those proteins, like the gene expression, during ocean acidification treatment, okay? So before I begin, I want to show you guys the paper real quick, because it's, wait, where's the mouse?
Where is
Wait,
that?
where's the mouse? The cursor.
Should be in there.
I don't see it at all. You guys see it?
Click it.
It was just there.
Just click it.
You were just doing something. I don't know.
Oh my God.
Maybe you need to move up here for it to work.
No,
Is it
it's
like
actually
too
the USB adapter is
No,
actually
it's
in the
on,
computer.
Oh,
Mhm.
sorry. Apologize.
Turn off and on.
Maybe you're just short
Battery.
Do you want to use mine?
Yeah. We'll try this first.
that should work.
It's like gone now.
Mm.
All the keyboard still works.
Maybe the battery is really low.
Alright, you guys, so I'm gonna use a keyboard. Look, before I begin, I just want to show you guys something cool. Like, this paper by Zhang et al., the inhibition of ocean acidification on the formation of oyster calcified shell by regulating the expression of these two random proteins, right? Well, I just want to point something interesting out, because the reviewer is none other than the famous Wei Xu from Texas A&M University.
I told you.
Corpus Christi.
I know them.
Yeah. And this is a cool paper because literally all the methods and the subject matter is basically everything that we do. So we'll see a lot of familiar stuff in this one. Okay, so let's get into it. Press this to make it full. How do I make it full on this?
The reviewer has left the chat.
Oh, I don't even know how to.
It's not touchscreen.
It is touchscreen.
Oh, it's touchscreen.
Yes.
Yeah, let's do
Oh,
that.
no
Leave it to
way.
the youngins.
Oh my god. It actually worked for me this time. Remember usually when I bump it? Okay, well, Dr. Xu left because he doesn't need to hear this, but he just doesn't want to hear this criticism. Right? So,
Here we go.
yeah. I'll get
You're
into
about
that
to be
near the
roasted.
end. Okay, it's actually good.
Really?
This paper is about oysters, it's about shell proteins, and it's about ocean acidification and gene expression. So we'll find a lot of this stuff very interesting and easy to understand. Okay, so just real quick, ocean acidification is just the process by which the seawater's pH is decreased because of increased carbon dioxide partial pressure in the atmosphere. Alright, so if there's more carbon dioxide in the atmosphere, then it will start interacting with the existing bicarbonate chemistry in the water. Okay, so increasing like one side of an equilibrium reaction between this weak acid is gonna start doing the Le Chatelier's principle and like causing more carbonic acid to form. Right, it's gonna move the reaction in that direction, and then the water's pH drops, not by much, just by a little bit, but still maybe substantial enough that it makes calcifying organisms have a more difficult time creating fewer, smaller marine calcifiers. Okay, so here's a picture that shows in better detail of a snail. I don't know what species this is, but as you guys can see, the snail shell is greatly reduced in terms of thickness, and shell abnormalities form because of ocean acidification treatment. Okay, so this is just an example. And so why does this happen? Well, there's a number of reasons, right? The first reason is shell dissolution because of the pH is lower. The second reason is because it's harder to calcify under this condition. And there's two reasons for that, right? The first reason is the interaction between the carbon dioxide and the carbonate chemistry in the ocean may reduce the amount of available ions, bicarbonate ions, to calcify. All right. The second one is the dynamic energy budget. Okay, this is an important term for you guys because we talk about it all the time, and now we finally know or I'm telling you guys what the name of it is. It's basically when your total energy in your body is limited, and so the allotment towards different activities is based off that limited amount. Okay, so if you have to suddenly start using some of that energy to maintain homeostasis somewhere, then it'll take away from the somatic and reproductive processes that also use that energy before. Okay, so if the conditions make it harder for something to calcify, in this case, basically calcifying organisms need to maintain a specific pH and bicarbonate concentration in the extrapaleo fluid next to the shell. And it's harder to do that when the pH is lower. Okay, so they need to expend more energy. And then now it affects the rest of their body processes because the energy is taken away from that. Okay, so dynamic energy budget. And the last reason, which is suggested by this paper, is due to changes in gene expression that regulate the shell formation. Okay, so we'll talk about this and whether it's actually true or not near the end. Right? Now let's talk about what they did. They take the oysters, right? They spawn them to make these trochophore larvae. This is 12 hours post-fertilization, so they're very young. Okay, they've only existed for about 12 hours so far. And then at this point, they're going to treat them in these 3 pH treatments. Okay, this is what we have right now. This is ocean acidification, and this is just some crazy pH level that was put in there just to get results. I'm serious. Okay, like I don't think we'll ever see this, and unfortunately, we only saw results when it was this low. Okay, so that's the first thing, right? What do we get? What are we looking at after this? The next developmental stage, which is the D-shaped larvae. You guys can't see the D because I think it's covering it.
You can move it with your hand. It's not
Oh, I can move it with my hand.
There's
Yeah,
a
that's
little
right.
arrow.
Ooh,
Yeah.
look at
If you
that.
click
fancy.
the little arrow above speaking, you won't see the videos anymore.
Oh,
You have
okay,
to go
okay.
over one
That's
hand.
fine.
Now we're never.
Alright,
gonna
so
do this.
does that look like a D?
Yeah, it does.
Yeah, it looks like a D, right? So
Oh.
that's why it's called the D-shaped larva, because the early shell, the first shell that forms, looks like a D. Okay, that's 24 hours post-fertilization. That means our treatment time was only 12 hours. Okay, so this is kind of reminiscent of the experiment I'm going to do next Tuesday, right? So basically, as soon as we get the larvae, we're going to treat them, but not with pH, with PAH, right? And then we're going to be looking at the shell
See what you
abnormalities
did there.
with the D-shaped larva. Okay.
Oh.
Well, before I continue, I want to take a look at this paper real quick, okay?
Because newspaper is published in the same place, Frontiers in Physiology.
Yeah.
Nice.
Ooh.
That's a good paper, huh?
And very likely one of their authors is going to review yours.
Yes.
Seriously.
Okay. So let's look at their methods, right? So we just talked about how they're gonna strip spawn to make the larvae, but look how they treat the larvae as soon as they finish. Triazole reagent. Look, we clearly already know what they're gonna be doing, right? Wait. Okay, so they're gonna be doing RNA stuff. Look, RNA extraction, cDNA synthesis. Familiar, right?
Oh my
Okay.
god.
Scanning electron microscope, they're probably using
Take
the
note of
Kingsville
this.
one. And
This is for my
qPCR,
paper.
this is literally
I just
the
wrote about all
exact
that.
stuff that we're doing, right? The only thing that I'm confused about is their statistical test,
Ooh.
the turkey test. I wonder which one that is.
Two keys.
Right, Dr. Xu, who reviewed this
Two keys.
paper?
Yeah.
Dang.
Yeah, because
Whoa, whoa, whoa.
I've never heard of that one before. Okay.
Hey, don't blame on the reviewers. I'm
Okay.
not supposed to give them typos and grammar
Okay,
mistakes.
yeah, that's correct. Right. But anyways, I know what they're talking about. Okay, so clearly this paper is really familiar with all the methods that we do in our lab. Okay, this is probably where Dr. Xu got a lot of the inspiration for some of their experiments as well, I think. Maybe not. Okay, no, maybe they got it from Dr. Xu, right? So what are we looking for we're looking for these two proteins, okay? The first one is called chitin synthase, and the second one is called chitinase. Now, if you guys know how proteins and enzymes are named, it basically works like this. If you have like some sort of chemical followed by synthase, then this is an enzyme that catalyzes the process of forming that chemical, okay? If you have a one word that ends in ase, then it's probably like a catalyst for a decomposition reaction. Okay, so for example, catalase decomposes hydrogen peroxide, and amylase decomposes starch, right? And here we have chitinase, which probably decomposes chitin. Okay, so why are we interested in chitin? The oyster shell is not made out of chitin, but it used to, right? Chitin is a very pliable and not super strong material. It holds structure, but it's not super strong, right? A good example is insect exoskeletons, which aren't super strong. You can squish them really easily, right? You can't squish like a seashell very easily, right? But you can squish an insect very easily because the shell is only made out of chitin, all right? Why we need chitin? Well, I made this analogy that chitin is the shell as cartilage is the bone. Okay, so if we know how the bone formation works in humans, right, you start with the cartilage and then the bone starts growing, it starts calcifying the cartilage, and it starts spreading throughout the cartilage until only the ends have cartilage, right? So like if this is the cartilage, the bone starts spreading out and only the ends now have cartilage, right?
I mean, like in a baby?
Yeah, in a baby, right, exactly.
Okay.
And this is important.
You're gonna describe the whole process of the long bone formation.
Yeah, long bone
30 seconds, go.
Oh.
No thanks.
No thanks.
Yeah.
You learned
what
it before.
process?
The long bone formation.
I
Remember that?
described it pretty well already. You know,
Yeah.
you have the cartilage and then it just starts calcifying and it grows outwards, okay? So that's what happens in the mollusk too, right? She just mentioned babies. That's true because we're talking about babies, right? The trophophore larva. And in the oyster trophophore larva, the first shell is just like the baby human. It's made out of chitin, right? So it's a pliable material because it needs to change shape, right? It's a D shape, it turns into clam shape and all that. And then that thing supplies nucleation sites that calcify into its calcium carbonate shell. Okay, so that's why it's similar to cartilage in our body.
So chitin is basically a protein. Is that right?
It is a protein. Yeah, so chitin is a protein. Anything that ends in IN is a protein. Even the word protein ends in IN, if you guys didn't realize. Okay, so,
Oh yeah.
yeah, so chitin, right? Anyways, the early shell is not hard, it's made of chitin so that it can calcify. Okay, so obviously these two things may have an important role in juvenile early shell development, maybe not the adult one, right? Okay, so let's see what they find, right? The first thing is they went on this crazy tangent. I don't know why they talked about phylogeny in this, right? But as you guys can see, they tried to figure out the phylogeny relating to other organisms of these two proteins, right? But I don't really like this, so I drew my own, right? Look at that, way better. Oh my, no.
What do you want to do?
No, how do I go back to the full screen? Oh, it works now.
Yeah.
Okay.
Uh-oh.
It's behind this thing.
This one?
Right there, right
Oh, you wanted to? Ah.
there. Oops.
Ah, now it works. Here. I got it.
Okay. Okay,
Did
alright,
you
so,
put Dr. Zhu on that?
yes.
Can't you find a better one?
No, this is the TAMUCC one. Okay. So, anyways, for the chitin synthase, it turns out that
Wait,
The-
Dan?
yeah.
Sorry, I can't see any of the presentation anymore.
Oh, okay.
Can you see me?
It's not sharing anymore.
Our online friends cannot see the presentation.
Can you go out?
Yeah, I'm trying.
Okay.
There.
Wait, where's the?
Oh, it stopped presenting. Sharing. Yeah.
Someone did.
Share.
Did you stop sharing?
Okay, thanks for stopping me, Molly. Because unlike the Mark retreat, they didn't
Use your
stop me,
imagination.
and they didn't see my entire presentation. It was so embarrassing.
Damn,
no.
no one
Yeah.
told you?
Yeah, I was so
Not cool.
angry at the end too. Like, oh.
This year. Or
But
next
yeah,
year.
thanks, Molly.
You're
Okay,
gonna
so
blow
anyways,
them away.
now that you guys can see this, the amino acid sequence comparison in chitin synthase, right? Turns out that the oyster, Crassostrea gigas, shares 44% amino acid sequence similarity to the Mediterranean mussel. Okay, it also shares 43% similarity to the fruit fly. It shares 41% similarity to none other than, what is this?
Zebra fish.
Yeah, Daniel Rario, right? And then it shares much less with these higher vertebrates over here. Okay. Well, this is really interesting for two reasons, right? One is, look at the zebrafish. It's a vertebrate. However, it seems to share a lot more in common with the invertebrates over here, okay? Second thing is, it's crazy that the oyster and the mussel, which are almost the same thing, are so far apart, right? Like, look at the difference between these things, 2%, 1%.
But that's only by looking at this specific chitin
Yeah,
protein
just this one, yeah.
conserving, that's why. Because
Yeah.
the zebrafish homology is like 69% with humans.
Right.
Yeah.
Yeah. I also want to show you a second reason why this number tricked me out a little bit. Okay, so 44% should not misled me a little bit to make it seem like it was far apart, but it's not, right? So just looking at this face value, it almost seems like, maybe, wait, where am I in the presentation? This is wrong. There it is. It almost seems like the zebrafish should belong in this upper quadrant, and then the oyster may be more derived than the mussel, right? But when we actually look at the amino acid sequence, see where the differences are coming from, then we see that's not the case. It looks like, see, the black ones are the similarities, right? So that means that most of the differences are in this part. I'm guessing it's probably some sort of uncoding DNA, like intron, something like that. That's probably,
no, those are all amino acids.
yeah, exactly.
So if it's amino acids, how can they be non-coding? It has to be coding DNA.
Really?
Coding means they code for the amino acids. So they've already transferred to the translated to the amino acids.
Oh, okay,
right?
yeah. It would have to be whole sequence.
Yeah.
Okay. Well, there are, I don't know. Maybe these differences are just not big enough, right? Or they're silent mutations in which even though you change the amino acid, it's the same. Oh no, that's not right. Yeah. Okay. Well, I'm not sure why, but addressing the zebrafish part, it does seem like the zebrafish is part of this group. See, the fourth one is the zebrafish, and it seems like it's part of the invertebrate group, the first three, right? It's very similar to those ones. It's very different from the other two. What we can get out of that is that I believe that the zebrafish has an archaic form of chitin synthase, that is more it's derived from the invertebrate lineage, and then the higher vertebrates are further derived from the zebrafish one, okay? So we could probably adjust our phylogeny to look more like this, okay? So you see how these are sister taxon right here with the zebrafish and the invertebrate lineage. They have the basal archaic form of this protein, and then the higher vertebrate line has adjusted it since then. Okay, and now it's different. Secondly, let's look at the chitinase, the second protein, and we see that this one is much more conserved throughout the rest of the animals. Okay, so it has not derived as much more than the chitin synthase has. Okay, I'm not sure the reason, but that is the data that we see. Okay, so real quick, I want to talk about this bird because they use this bird to put right here. Okay, so since we're on a tangent anyways, I might as well take it here, right? I just want to show you this because it's cool, right? Look at its scientific name, Cyanistes ceruleus. Can anybody guess what color this bird is supposed to be?
Cyan.
Blue, right? It's literally has blue twice in its name. Cyan and cerulean, right? That's a, I don't know, that's a strange name in my opinion. But
Wait,
yeah,
but
this
even the
bird
moss it's sitting
is-
on. Oh, I was gonna say this is a black and white picture that
I
you
did it on
sent
purpose,
to us.
right? Yeah.
Detective Sandra.
Yeah, so anyways, it's a bluebird, right? I'm not sure what exactly, it's a type of tit. Yeah. Okay, so let's go back to the actual interesting part, right? Maybe by now we've even forgotten what the paper is talking about. I don't really know why they stuck that phylogeny section in, but the results of the experiment go like this. They found that, can we move that real quick? They found that if you decrease the pH, not surprisingly, shell deformities occur, and we see incomplete calcification of the shell. Okay, so look at this one. This one's so smooth and completely calcified. It has formed a nice D-shaped shell. Okay, now look at these ones right here. These ones have fissures and indentations showing spots in between calcified portions that have not calcified fully. Okay, so incomplete calcification is definitely occurring. It is definitely harder to calcify during these low pH conditions. Okay, now let's look at the chitin and synthase and kinase. Chitin synthase on the left, kinase on the right. We notice that there's some sort of trend that's going on here, okay? And the trend that was identified by this paper is this. Increasing pH causes chitin synthase to be downregulated, whereas increasing pH causes chitinase, the chitin destroying one, to be upregulated. So if that is the case, then the paper has come up with this statement, that shells are reduced during ocean acidification, not because of the other two reasons, but because chitin synthase, the formation of chemical, is downregulated, and chitinase, the destruction chemical, is upregulated. Isn't that crazy? It's almost like the animal is halting the development of a shell and even destroying it during ocean acidification. Okay, well, let's look at what other support they have. To look at the gene expression, a distribution around the body, I don't even have to highlight this, it highlights itself. It shows that basically the protein, the chitinase and chitin synthase is upregulated right along the margin of the shells. You can even see the bivalves right there, right? Look at that, right at the margin, right where the growth is occurring. All the growth of the shell occurs at the margin because that's the part that gets elongated, right? It doesn't grow in the middle, it grows at the very edge. Okay, so that happens for both chemicals, and comparing to the control, in which there's no concentration point of either chemical, right? It's concentrated here in the edges, but here it's all around the bodies. Like, see all those dots, right? There's no specific concentration site. It shows that indeed both of these chemicals are differentially expressed during ocean acidification, right? That is definite, right?
Yeah,
It
so
is
this is
confirmed.
actually the pretty useful technique. So Kenzie, you probably wanted to talk to Dan Wagner about the methods. So this is a VCU hybridization, so allow you to identify the certain mRNA, the location, the
Yeah,
expression
the location, right?
you
of
are
those
interested.
cancers.
So very cool technique.
Yeah, so it has confirmed that both of these are indeed involved in up and down regulation, differential expression during ocean acidification. Okay, but here's one thing though, I'm not really sure about this. Okay, I'm not really sure about this trend here, because let's just pay attention to the D-shaped larva. Look what I see. I see that increasing pH actually upregulates the chitin synthase, right? I'm not sure if I agree with the original interpretation. Look at the early D-shaped larva. It doesn't even change the chitinase with the increasing pH. They kind of just picked and choose, right, to fit their narrative. Look at this. The chitin synthase only decreased in the early D-shaped larva. In the control, it didn't change at all. And in the, the trophophore larva that's not forming the shell didn't change at all. And in the D-shaped larva, it actually increased. Right, so that's conflicting. We shouldn't trust that. And over here, everything was not significantly different unless we're talking about the D-shaped larva, which it did actually increase. Okay, so rather than listen to that, right, this is their explanation. Rather than listen to that, I would like to offer my own explanation, right? If we read this closely, basically all it says is exactly what I said. Chitin synthase goes up, or goes down, and chitinase goes up, and there we go. There was no explanation offered and as to why. That was just what they put. I feel like they could have put a little bit more work into trying to explain the discrepancy. Maybe try to explain my observation here, but they didn't, right? I think that part of the paper is a little incomplete. So let me offer my explanation, right? Looking at this, in which, first of all, chitin synthase increases with the D-shaped larva through increased pH, which is contradictory to the original statement. Chitin synthase does decrease, but for the early D-shaped larva only. Okay. Chitinase increases in the D-shaped larva, as they initially mentioned. However, in the other life stages, it doesn't do anything significant. Okay, so it's hard to trust the original things. Why do I think this is the case? Because if you follow shell formation from the very beginning, it probably works like this. Ocean acidification inhibits the chitin synthase activity in the early D-shaped larva when they're trying to build the shell. Okay, it's a stressful situation. The dynamic energy budget probably makes it so that early D-shaped larva has a hard time making a shell and inhibits the production of chitin synthase. Okay, but then now, as it is developing into the D-shaped larva, all of a sudden it wants to build a shell now. Okay, so I'm guessing it's some sort of hormonal change that causes it to start to try to build the shell. It starts upregulating chitin synthase to build the shell during the D-shaped larva stage. Okay.
That's a kind of one of the hypotheses we have for our NSF grants. So we're also trying to looking at the potential like signal for what initiate the build of the shell at a certain point.
Yeah.
So my original thought is probably the calcium signal. So the calcium signal, my opinion is in the very young stage, they're probably not that high concentration in their cellular level, but at a certain point, it start to, a lot of the channels start to open up. They start to absorb the calcium into their body and start to build the, the cell on the sudden.
Yeah.
And, that's the whole 5 year of the project. So we were really interested in knowing that.
Mm-hmm.
Yeah.
All right, let me finish this real quick.
Mm-hmm.
So shell formation is accelerated in the D-shaped larva all of a sudden, and so it starts to try to upregulate chitin synthase. However, remember how ocean acidification is going on? It may be difficult for it to develop a shell. So as it is upregulating the chitin synthase, it produces an excess chitin that doesn't get used because it doesn't get calcified because it's so hard. And so because of that, I think that the reduced calcification by ocean acidification causes the animal to want to resorb the excess chitin that it created. And so it upregulated its chitinase in that process. See, this is probably a slightly more complete explanation, even though it's not backed up at all. I just made it up, right? But it's I don't really buy the original explanation though, that the shell formation is decreased because it didn't produce enough chitin synthase and because it started destroying its chitin. Now, I think it was just decreased by the original methods: shell dissolution, dynamic energy budget, and difficulty calcification. And then as a result, this followed. Yeah, and I also think the changes in regulation of chitin synthase has nothing to do with ocean acidification. I think it was merely hormonal.
What would you do to test that? To test your theory or to test?
I would look at maybe no acidification and see if it increases levels.
If you're interested, there's in 2012, there's a Nature paper. They did a whole lot of sequencing work, sequenced the individuals of the larvae all the way from trichophore stage to sediment stage. So they compare their gene expression. So if you're interested, you can dig out all those data to see if there's anything over differentially expressed during between and after the formation of the shell. Maybe we can find something like what you said, maybe hormones or something.
Yeah.
Differentially expressed.
Oh, okay. Actually, I misspoke earlier. Sorry, Kenzie. So, I was wrong. chitin synthase was affected by the acidification, but it was affected in a way of like a compensatory mechanism. So remember how the early trochophore or early D-shaped larva was downregulated chitin synthase? I believe that downregulation had to be made up at a later stage by the D-shaped larva. That's why it increased. Sorry, it was not because of what I just said earlier. Yeah, because the control one did stay flat through the pH. But yeah, I think the early trochophore larvae did have some difficulty. So with the inhibition of its chitin synthase, and the chitin synthase had to be compensated during the D-shaped larva stage. That's why it went up. Yeah. Okay. So, anyways, that's the end of my presentation. Hopefully, it was interesting.
Great.
That's interesting, thank you.
Any questions?
I don't think so.
But make sure I wrote it right. What process did you want me to go over with Dan? That